Viral genetics. Probability is important in the survival of virus in nature. The capacity of replicate is the capacity to replicate rapidly. But if replication is too rapid, it can be self-defeating. The capacity to replicate rapidly, but if the replication is too rapid, it can be self-defeating. The capacity to replicate to high titer, like arbovirus, which give very high vertebrate host viremia. The capacity to replicate in certain key tissue, the capacity to grow in immunological sequestrated site, like brain, kidney, or in the cell of the immune system. The capacity to be shed for long period of time, intermittent shedding, is an additional advantage to the virus. And intermittent shedding is additional advantage to the virus. Define viral diarrhea, infection, and cattle. The capacity to elude host defense. Virus, especially those with large genomes, have genes that encode proteins capable of interfering with host antiviral activity. Chickenpox virus immune tolerance is another mechanism that combines viral diarrhea. Cattle capacity to survive after being shed into the external environment. Canine parvovirus has environmentally stable capsid. The capacity to be transmitted vertically. Recombinant DNA technology, bacteria and plasmid isolated and gene inserted into the plasmid. After that, the plasmid is put in the bacterial cell. The cell multiply with gene for interest, and cortisol protein is produced. Protein used to make snow from a higher temperature. Protein dissolve blood clots in the heart attack already. Gene used to alter bacteria for cleaning up toxic waste. Gene for pest resistance inserted into plants. This is the application of this recombinant DNA technology. Polymerase chain reaction. This is the PCR, and this is its step. First, DNA is denatured. Then DNA is denatured. Continued round of purification. Another round of denature. Primer are attached, and the number of DNA that are doubled. Mutation. Definition error in the coding of the viral nucleic acid that occur during replication are called mutation. However, a particular mutation is lethal to the virus. It depends on the result. Phenotypic characteristic virus plague technique could be used for studying and isolation of the virus variant. This is the plague assay. Types of mutation. Genotypic mutation, also called a base substitution, occurs when a single nucleotide is replaced with a different nucleotide. A point mutation results in a base pair substitution after replication and mostly a mutant protein after transcription and translation. Parental DNA point mutation, here difference in one base pair. And this is the normal daughter DNA, and this is the mutant daughter DNA, and here the substitution of the first pair. A genotypic Insertion mutation, in which extra base pairs are inserted into a new place in the DNA. A deletion mutation, in which a section of DNA is lost or deleted. Base substitution. Two main types of base substitution: transition purine or pyrimidine to pyrimidine, transverse purine and pyrimidine or pyrimidine to purine. Transition are more insertion and deletion of deletion. Insertion or deletion of one or two base change the reading frame. A frame or shift mutation error occurs at the DNA level, causing the codon to be parsed incorrectly. Missense mutation, in which substitution of a single base pair results in codon change, thus giving a single AA substitution. Phenotypic changes. Condition lethal, multiply under some conditions but not others. Wild types grows under both sets of conditions. Temperature sensitive mutant don't grow at higher temperature. Host range mutant don't grow in all cell types that the phenotypic changes. Like a mutant may produce a different type of plaque in a cell layer. Natural weak Rift Valley fever virus mutant. Antibody escape mutant may become resistant to neutralization by antibody raised against the wild type in host persistently infected with infectious anemia virus. A prominent example of mutation affecting viral antigenicity is the antigenic drift of influenza virus. Phenotypic changes of mutants grow better at elevated temperature than less susceptible to most fever response. Attenuated mutants milder, no symptoms, vaccine development, pathogenesis. Defective interfering virus demonstrated in most families, especially RNA virus, and need the presence of the wild type virus. Mutagenesis, spontaneous mutation, error in replication, transitions, and transversions that occur during replication and can result from deamination of the cytosine. Here, the cytosine loss of amino group become uracil. Spontaneous lesions, deburination, deamination, oxidative damage to DNA. Induced mutation based analog mutagens from uracil can substitute for C and then one up binding with G result in miscoding. The example, bromuracil AZT, a nucleotide analog that can result in a chain termination. AZT. Induced mutation, chemical agents such as nitrous oxide or nitrosoguanidine, ultraviolet ray, ionized radiation, cross-link between adjacent dimers, site-directed mutagenesis. The nucleotide at any prescribed position in a DNA genome or Complementary DNA not transcribed from an RNA genome can be changed to any other nucleotide, or it can be deleted or affected by the early insertion of an additional nucleotide. It can be done in bacteriophage vector M30. And here gene is the vector, and here DNA polymerase, and here transformed, and here the E. coli host cells, and here the cell division. Genetic change according to some processes like mutation, recombination, exchange of information between two genomes, interaction between viral gene products. This may involve not only the nucleic acid but also the proteins. The products of such interaction may affect the viral genotypes. Recombination, intramolecular recombination. It involves the exchange of nucleotide sequence between different but usually closely related viruses during replication in the coronavirus, and the Toga virus. Common DNA viruses. Recombination, genetic reassortment. Observed in RNA viruses with segmented genome, Bonnier variety, Festival fever, and Osmotic variety, and blue tongue influenza virus. Exchange of segments may occur when cells are infected, sometimes with two related viruses. Genetic change and recent evidence of viral evolution mechanism, example, point mutation, avian influenza, intramolecular recombination between West Nile and Alpha virus, genetic reassortment, pandemic human viral human influenza H1N1 in 1918, originated by genes from avian viruses, intramolecular recombination and mutation, poliovaccine virus, reversion to virulence following vaccination. Recombinant DNA technology. Box as a vector and recombinant DNA technology make vaccine. The large genome of virus have allowed the insertion of large amount of foreign DNA into non-essential genome regions. Okay. And there is veterinary vaccines prepared for vaccine. Recombinant vectors are generated by incorporating the foreign DNA into viral genome via homologous recombination. Proteins are expressed properly, have undergone post-translation modification. Recombinant vaccine virus for use as rabies vaccine. Vaccine virus for rabies vaccine. And the raccoon eating bait with rabies vaccine in it. The future of recombinant virus technology, we can make multiple vaccines in one shot. Practical achievement of recombinant DNA technology: complete sequencing of genome of the presented virus of all the families of DNA virus containing animal pathogens, characterization of the virus or proviral DNA that are integrated into the chromosomal DNA of transformed cells and animal infected with retroviruses, development of probes for use in rapid diagnostic assay, including hybridization assay, application of PCR to amplify specific viral genome sequence of diagnostic and research purposes, production of protein coded by specific viral gene using bacterial, yeast, bacteriovirus, and animal cell expression system.